So in this video I will discuss on sugar derivatives or the derived monosaccharides. Now these sugar derivatives, they are similar to the monosaccharides, but they differ in regard to aldoses and ketoses. And some of the important derived monosaccharide, the sugar derivatives, are sugar acids, sugar alcohols, then sugar phosphate, deoxy sugars, and amino sugars. These are some of the important derived monosaccharides. Now let us take a look at each type one by one. So what are deoxy sugars? Now deoxy sugars, they are formed when a hydroxyl group is removed and it replaced by a hydrogen. It results in loss of one oxygen atom in a molecule. So such sugars, they are called deoxy sugars because they lack oxygen. Okay, so deoxy meaning lack of an oxygen atom. So these type of sugars, they are referred to as the deoxy sugars. Now if you take a look at the example here, that is the ribose sugar. Okay, the D-ribose sugar, this is its structure. But now from the ribose sugar here, if you see at carbon number 2, the OH group is replaced by a hydrogen. That means in the second carbon atom there is a lack of oxygen molecule. You can see here also hydrogen and here also hydrogen. But in case of ribose, we have the hydroxyl group OH. So due to the lack of oxygen atom here, this molecule is now referred to as the deoxyribose. Alright, this is because the oxygen is absent in carbon number 2, so it is referred to as the 2-deoxyribose. Now there are some other sugar molecules where the oxygen atom is absent from the hydrogen, from the second carbon atom as well as the third carbon atom, as you can see here in this example. So this molecule will be called as the 2'3' dideoxyribose. Alright, it is known as dideoxy. Di meaning two oxygen atoms are absent, so dideoxyribose sugar. So this is the example of the sugar derivatives, that is the deoxy sugars. Now this molecule here, the 2'-deoxyribose, is an important constituent of DNA molecule. It has the oxygen removed at carbon number 2, and dideoxyribose, which has a carbon the oxygen atom removed from carbon number 2 and carbon number 3, it is used in DNA sequencing. So these two deoxy sugars are very important. This is found in the DNA and this molecule is used in DNA sequencing. So there are other examples, okay, other example of deoxy sugar molecules, and those are L-fucose. Alright, we have L-fucose and we also have L-rhamnose. Okay, so this L-fucose and L-rhamnose, they are deoxy sugars. They are hexoses. Okay, hexoses, that means they have 6 carbon atom molecules. But these deoxy hexoses, they are formed when the oxygen, it has been removed from carbon number 6. Alright, that means from carbon number 6 the oxygen has been removed. From D-galactose and L-galactose and L-mannose respectively, we will get these two molecules. So another example of sugar derivatives, that is of deoxy sugars, are L-fucose and L-rhamnose. This is formed due to the removal of oxygen atom at carbon number 6 of L-galactose. And this other one is formed from the removal of oxygen atom from carbon number 6 of L-mannose. Okay, so we will get these two sugar derivatives, and these, therefore, they have a CH3 group at carbon number 6. Alright, these two, they will have a CH3 group at carbon number 6 and not CH2OH. Why? Because the oxygen atom, it has been removed, so we get CH3. Alright, so in these sugar derivatives, which are derived from these two sugar molecules, two monosaccharides, due to the removal of oxygen atom in their carbon number 6, they will have CH3 group and not CH2OH. Now we come to the next another type of sugar derivatives. They are amino sugars. So next here, the amino sugars, how they are formed. Okay, it's when an amino group may replace a hydroxyl group. Remember this. Okay, the amino group, it may replace a hydroxyl group at carbon number 2 to produce amines of glucose, galactose and mannose. As I had shown here in this figure, you can see from the glucose molecule, if the hydroxyl group at carbon number 2 is replaced by an amino group here, you can see, then we get beta D-glucosamine. Okay, we get amines of glucose, galactose and mannose. Here in this case, galactose, replacement of hydroxyl group in carbon number 2 and also in carbon number 2 of mannose. Okay, so we get the amines of these three monosaccharide. Now another reaction, like further addition of these molecules, these amines, these amino sugars, they may be further converted to other complex forms by linking an acetyl group to the amino group. Okay, by linking the acetyl group to this amino group, we get a more complex forms. Now those complex forms, as you can see here, if an acetyl group is attached to the amino group here, we get N-acetyl glucosamine, N-acetyl galactosamine. And the lactic acid molecule, it may condense with, you can see here, this is a lactic acid molecule. Okay, it may condense with the hydroxyl group at carbon number 3. Okay, at carbon number 3 of glucosamine, and thus forming muramic acid. So what happened here in glucose molecule, if at carbon number 2, if it replaced by an amino group, then we get amines of glucose, right? Beta D-glucosamine, as you can see here. But then if it further linking of an acetyl group to this amino group, we get a more complex form, that is N-acetyl glucosamine. Now, but in this glucosamine, if the lactic acid molecule, okay, it may condense with the hydroxyl group at carbon number 3 to form muramic acid. So these are the example of the amino sugars, okay, the derivative of monosaccharides. saccharide molecules termed as the amino sugars. So the next one we have the sugar alcohols. Now in sugar alcohols, the aldehyde group of some monosaccharides, they may be reduced to alcohols. This one you have already studied in the reaction of monosaccharides. Like this is a reduced reaction where the product form they are the polyhydric alcohols, okay, which produces sugar alcohols due to the reduction reaction of monosaccharides. Now you can see that the aldehyde group of monosaccharides, they get reduced to alcohol. So you can see example here in case of glucose, where this aldehyde group it gets reduced to alcohol, and also the product form is referred to as sorbitol. Okay, this is one of the example, and the second example of sugar alcohol is mannitol, which we obtain from D-mannose, from the reduction of aldehyde group here of mannose sugar. Okay, so you can see now we get the both end of the chain, they are similar. We get alcohol group in both the end of the chain, same in case of sorbitol. So these are known as the sugar alcohols. Next we come to the sugar acids. All right, so we come to sugar acids. Now sugar acids, okay, the monosaccharides, they may be oxidized, or they may be oxidized to acids, okay, at either carbon number 1 or at either carbon number 6. Now the important ones are beta D glucuronate, okay, where at carbon number 6 the CH2OH group it gets oxidized to carboxylic acid, okay, COOH. And another example is D-gluconate. Here the aldehyde group at carbon number 1, it gets oxidized to carboxylic acid. So these sugar acids are obtained due to the oxidation reaction of monosaccharide molecules. So they are also termed as the oxidized sugars. Okay, the first one, when we obtain sugar alcohols, those molecules are also referred to as the reduced sugars, and these are termed as the oxidized sugars. Now one of the example I can show here is so you see here this carbon number 6, the alcohol, it gets oxidized to carboxylic acid. So this molecule is termed as beta D glucuronic acid. Okay, and the another example okay, so another example, as you can see here, the carbon number 1 Here the carbon number 1 of a sugar molecule, it gets oxidized to carboxylic acid. This is termed as the beta-D-gluconic acid. Okay, so remember these two examples, the glucuronic acid where the carbon number 6 it gets oxidized, and the gluconic acid where the oxidation it takes place at carbon number 1. And another, that is the last important group of sugar derivatives, okay, it is the sugar phosphates. So the sugar phosphate here, the monosaccharide, they may be phosphorylated, usually either at the first carbon, as you can see here, this is the first carbon, or they are phosphorylated at carbon number 6, okay, or sometimes at both carbon number 1 and carbon number 6. Now glucose 1 phosphate, glucose 6 phosphate, and glucose 1,6 phosphate. As you can see, there are others like fructose 6 phosphate, ribose 5 phosphate. All these, they are the phosphorylated sugars, and these type of phosphorylated sugars, they are of great importance in metabolism. So you can see here that in glucose 1 phosphate, the 1 here, it indicates the number or the carbon number, okay, the atom, carbon atom number 1, it gets phosphorylated, whereas in this case carbon number 6 it gets phosphorylated. So here we have glucose 6 phosphate. When both carbon number 1 and carbon number 6 they get phosphorylated, so that molecule is named as 1,6 bisphosphate. Okay, so this is a glucose 1,6 bisphosphate. And we also have fructose 6 phosphate, ribose 5 phosphate, and so on. Okay, so these are the sugar phosphate derivatives of monosaccharide molecules. Okay. Next in this video, I would also like to talk a little bit on these glycosides. Okay, what are actually glycosides? Now glycosides, they are those organic compounds from plant and animal sources. Okay, they are organic compounds from these sources which on enzymatic hydrolysis they give one or more sugars, they give one or more sugar moieties, okay, one or more sugar moieties along with a non-sugar moiety. Okay, that means glycosides, they contain both a monosaccharide and a non-monosaccharide molecule. Okay, they consist of both monosaccharides and non-monosaccharide molecules. And here the common sugars found in the glycosides, okay, because just remember that glycosides, they have two parts, the sugar and the non-sugar part. So the sugars which are mostly found in glycoside molecules, they are glucose, then we have mannose, rhamnose, and also digitoxose. Okay, so these are the 4 types of sugars commonly found in glycosides, whereas the non-sugar part, which the common non-sugar part which are found in glycosides, they are the alcohols, then we have the phenol and the amines. Okay, so you can see that glycosides, they are not actually carbohydrates because they consist of a carbohydrate part and a non-carbohydrate part. Okay, and here the carbohydrate or the sugar part is referred to as glycon, whereas the non-sugar part is referred to as aglycon. Okay, so the activity of glycoside is due to this non-sugar part, okay, is due to the non-sugar part, whereas this sugar part of glycosides, they plays a role in the other properties of glycosides like absorption, then metabolism, distribution and excretion. Okay, so both these parts, they have different activities, different functions. So I just want you to know a little bit on this glycoside molecules, that they are not actually pure carbohydrates, but they consist of monosaccharide and non-monosaccharide molecules. Now, what are the importance of these glycosides? Many glycosides, they are usually occur in plants, often as the flower and fruit pigments. Okay, they occur in plants usually as fruit or the flower pigments, and the common example of pigments found in plant is anthocyanins. Okay, so these are nothing but they are glycosides. And another important of glycosides is that they are very important in various medicines. Okay, the medicines, then we have condiments, and also dyes from the plants, they occur as glycosides. Okay, so condiments and dyes also which are present in the plant, they occur as glycosides. And we have these glycosides which are of great value are the heart stimulating glycosides. Some glycosides are known to stimulate the heart, and those are obtained from digitalis, okay, and also from strophanthus. Okay, these members of a group of the glycosides which are obtained from digitalis and strophanthus, they are the heart stimulating glycosides, and they belong to a members of a group of glycosides known as the cardiac glycosides. Okay, because they can stimulate they have the stimulating property, they can stimulate the heart. And many other several antibiotics, okay, the antibiotics such as the streptomycin, okay, such as the streptomycin, also streptomycin, they are nothing but they are glycosides. And we have saponins. Saponins, they are widely distributed in plants, and these saponins also, they are glycosides that lower the surface tension of water. And saponin solution, they have been used as the cleansing agents. And then we have glycosides which are derived from glucuronic acid and steroids. They are the constituent of normal animal urine. And also the compounds or the nucleosides derived from the partial breakdown of the nucleic acid. Okay, these are all glycosides. So we see these are some of the importance of the glycosides.